Okay, so welcome to a sheep brain, and so here we can see that this is the sheep brain and it is different from the human brain. Can you please pass me the human brain model? In the shape of it. We can see here that it's on a very horizontal plane, but this is the human brain and we can see how it's more of a C shape. But it's made up of the three same parts. We have the cerebrum, which is the largest part, we have the cerebellum, and we have the brain stem. And on the human brain we have the cerebrum, the cerebellum, and the brain stem. But the reason why we've got changing is because we're bipedal and this sheep is a quadruped. So let's talk about protection for the brain. The brain, the first part is that you've got this lovely big hard skull that goes around the outside of the brain, enclosing it and making sure that there is no damage that can be done to it. Underneath that you have three meninges. Meninges are the membranes that go around the outside of the brain. The first meninges is called the dura mater, and the dura mater, you can't actually see it on this brain because it's been removed, but here's one I made earlier, and this is a sheep's brain that has got the dura mater on it. It looks like a swimming cap and it's quite thick, and you can see that it is quite a thick substance. This dura mater and these meninges would actually travel down the entire of the spinal cord, wrapping it in there. So the first layer is the dura mater. It's the closest one to the surface of the skull. Underneath that you have the arachnoid mater. The arachnoid mater is called the spider-like a spider mother, and the reason why it's called spider mother is because it looks spidery, web-like. Now the dura mater, it roughly translates to meaning tough mother, and it's got a good name for it because again it looks tough, and the arachnoid mater looks like spider. Below that you have the subarachnoid space, and that's going to allow for blood tissues, and you can have CSF go through there as well. And then here, if we look on the surface of this brain, you can see that there is some, oh, hair in my face. You can see there is some what looks like saran wrap, a thin plastic coating that is over the surface of the brain. I don't know if you can see it or not, but here you can see, particularly on this section right here, that, shall I bring it closer? That right here, if I give it a wiggle, you can see, oh, you can see it lovely right there. You can see this very thin layer that is shiny and clear looking. That's the pia mater. The pia mater means tender mother, and that goes around the surface of the brain. So if I was looking at this, I would want to find the left the left hemisphere, I beg your pardon. The brain is divided in two hemispheres. I'm looking for the left. It would be this one located under my finger. The reason why this is the left is because this is the anterior portion of the sheep and this is the posterior portion of the sheep. So in our drawing we would label this as we're looking down on it, so this is the dorsal view, this is the anterior, this is the posterior, this is the dorsal, and underneath is the ventral. We can see also that the cerebrum is divided into two hemispheres. These two hemispheres are divided into four lobes, which we can actually see clearly on this model as well. In the front we have the frontal section right here, the frontal lobes. They're on both sides of this hemisphere. Then on the sides you can see that there's a bit of an indent. That is going to be the temporal lobes, and the temporals are going to be on the sides here. You think you've got an indent where your temples are. Then on the very back here, this is the occipital lobes, occipital being for vision and sight. And then in the middle between the frontal and occipital is the parietal lobes up here. Each lobe has its own function. Again, the cerebellum is back here and the brain stem. The function of the cerebellum is going to be for coordinating balance and movement, and as a relay station to get everything to the rest of the brain. This line down here is really important. This is the longitudinal fissure. The longitudinal fissure is going to basically divide it into two hemispheres. But if we open it up and we look down in between it, we will probably see the corpus callosum. Oh, I'm just trying to break some of this pia mater. You can see some of the fibrous connective tissue. That's going to be part of the corpus callosum, which is going to be the connection between the two hemispheres. We'll come back to that in a minute. Okay, so again, frontal, parietal, occipital, temporals, cerebrum, the longitudinal fissure. We have the different four lobes we talked about. Then we have the cerebellum and the brain stem. We're going to have a look now at the one of the structures inside. So as I pull it down, oh, we got cut by accident. Here we go. As we pull it down, you may notice, as we look up inside, we're looking anteriorly inside, we can see what looks like a bottom sitting on a toilet seat. And then there is a knobble right there. Hopefully you can see the knobble. Okay, there's the knobble, here's the bottom, here's the toilet seat. And there is an important factor with those. Okay, so, oh, get to this side, because I have to spell it. All right, so this is the bottom, oh, I lost my thingy here. The bottom is the the bottom sits on the toilet seat. The bottom is there for response to a visual stimulus, and that is going to be, if you see, let's say, a flash going this way, you turn to look at it, that's going to be the superior colliculi. I actually like calling it the colliculi, but that's me. The bottom is going to be if you hear a sound, it makes you jump, just like my dog did, and you turn to look towards it. That's the inferior. Remember, superior means above, inferior means below. And that little knobble that sits up there, that's really important, because that's the pineal body. And the pineal body is going to be there to secrete melatonin. Melatonin is useful for the production of sleep. So melatonin is going to increase in levels as the light decreases, and as the light decreases, melatonin production increases and it makes you feel sleepy. The problem is nowadays that we have all of these lights that are bombarding us with cell phones, with televisions, with computers, and these blue lights in particular really mess up with our production of melatonin, and so that's why we have a lot of people that have difficulty sleeping. Okay, we are going to now flip over and have a look at the ventral side. Make sure you draw this. Okay, on the ventral side, we're going to start at the very front with my birds chirping in the background. You may be able to see these little flaps at the very, very front of this ventral anterior portion. They're lighter in color, and if I use my probe, I can actually move it just slightly. This is the olfactory bulb. And there are two of them, the olfactory bulbs. Olfactory for the sense of smell. Olfactory sense of smell. So this sense of smell is there to help us with deciphering odors. Now, humans have one of the worst and smallest sense of smell. Dogs are significantly much better because they go around using sense of smell and pheromones and the like to communicate. Thankfully, we don't go around smelling each other's bottoms to communicate. Otherwise, if we did, our olfactory bulbs would be significantly large, like dogs, sheep, and whatnot. Okay, so another sensory device that comes in is at this location right here. This is the optic chiasm. It's a cross, and that's because the optic nerve comes in from the two eyes and it crosses over here. So we can actually see that a little more clearly on this brain here, because these are eyeballs. And you can see this is the optic nerve coming off of the eyes, and it does a little crisscross right here, and this is the optic nerve. This is where this eye is going to be processed by this side, this eye by this side. So they get switched. So my left eye, if I'm just opening my left eye, it's being looked back here and vice versa because of this optic chiasm. Now, very important structure right underneath or below right below the optic chiasm is this hole where my probe is in. The hole is called the infundibulum, and this is where the master gland used to be located, the pituitary gland. In fact, the infundibulum is where the stalk of the pituitary gland would sit, and you would then have that gland sit there. A very important gland secreting hormones again. This is the mammillary body, and then we get into the midbrain and hindbrain. So this is the midbrain, and then this comes towards more to the hindbrain. This is the brain stem. We'll work backwards from the posterior towards the anterior. This is the brain stem. So this would be connected to the spinal column, and all of the other nerves would come in, get passed up through here. Additionally, this brain stem has got two ridges. You've got the pons and the medulla oblongata. The pons is in the anterior position, and the medulla oblongata is closer towards the back. So this is the medulla and this is the pons. The medulla oblongata is a region that helps with autonomic function, so breathing, heart rate, blood pressure and the like, regulating your sleep-wake cycle. And the pons is again very important in the same sort of thing, sleeping, breathing, and as a relay station to transfer everything that was coming in through the spinal column to the rest of the brain. Okay, I think that we've covered everything on here. What we're going to do now is we are going to cut the brain, and we are going to cut it down the longitudinal fissure, and oh no, mosquito. And we're going to make sure that we cut carefully. So I'm left-handed, so I want to make sure whenever I'm cutting and dissecting that I keep things the way that it's best for me. So I'm going to hold it with my stabilizing hand, keeping sure that my scalpel is kept out of the way. I'm going to run my scalpel down the longitudinal fissure, and I'm going to cut through where the connection is, which is the corpus callosum. I'm doing a gliding motion with my scalpel. I'm looking where I cut so that I don't cut too many structures. Now I'm going to continue that imaginary line down through the cerebellum and down the brain stem. Oh, we're getting there. Looking where we cut. Few more incisions, and then we have it in half. Excellent, very good. So we will take a picture of this so that you can then draw it properly, and I like this side better. Here we go. right, so now we're going to look at the internal structures of the brain. One of the things that we noticed on the outside of the brain that we didn't discuss were the ridges, which we can see very clearly on here as well. These ridges and grooves increase the surface area, which increases neural cognition and makes us have a higher order thinking. Now, the wrinkles, the gyri are the ridges and the sulci are the valleys. So ridge and valley. This increases the surface area, and we can see that coming along the outer edge of the internal cut here. One of the biggest things that most people don't realize is you have a hole in your brain, quite a big hole. This hole is called the lateral ventricle, and this lateral ventricle runs quite deep. It's not superficial, it's a deep hole, and the inside of it has some very special cells. These cells generate CSF, which stands for cerebrospinal fluid. This is the fluid that goes inside the brain, around the outside of the brain, down the spinal cord, and back and around. It works as a cushioning, cooling, and CCC, cushioning, cooling, and circulation for the brain. Okay, CCC, that's how we remember it. Cushioning, cooling, and circulating for the brain. So the CSF is generated in this lateral ventricle. It then comes out and it goes down this way, down the aqueduct, over the top of the bottom, and then underneath the cerebellum, down the brain stem, back down and up and around, and it goes around the outside. So your brain is cushioned by that CSF. It's like hydraulics. It helps to prevent that from squishing. Okay, so then this region right here, hopefully we can see, this region right here, which is running on top of the lateral ventricle. This part right here, that is going to be the corpus callosum, and that's the connection between the left hemisphere and the right hemisphere. So if somebody is very artistic or linguistic, or somebody's very scientific and analytic, they may be using different sides of their brains. And we tend to have connections between them. These connections are the bridges. We tend to have 5 to 7 bridges per person, but it was Einstein who actually he found and interviewed the person who was going to do his brain autopsy, and they found when they did the brain cuts that Einstein had over 20, like 25 different bridges in his corpus callosum, allowing him to utilize more of his brain simultaneously, thus enhancing his genius. Okay, so one more time, let's keep going. So we've got the lateral ventricle here. You may see what looks like a circle, like a thumbprint right here. This thumbprint, again, this is going to be the thalamus. And we also have the hypothalamus, which sits just below it. We can see also the optic disc, right, or the optic nerve, I suppose, right here. But this is the thalamus. And the thalamus, pineal body, and then where the pituitary gland was, you can see in this midbrain region there is a lot of endocrine production, hormone production, endocrine use. So we have a lot of hormones being generated by the brain in the middle here. Oh, bird. That was my inferior colliculi. I heard a sound and I turned to look. There we are. Okay. Pineal body, colliculi, thalamus, hypothalamus, the infundibulum, the pituitary gland, pons medulla, so relay station. We're coming to my favorite bit right now. Look how pretty this is right here. This is the inside of the cerebellum, and it is rightly called the tree of life, the arbor vitae, which is a difference between the white and the gray matter that is being shown inside this brain. The cerebellum, again, is going to be a relay station, but very much involved in balance, movement, coordination. And arbor vitae, arbor meaning tree, vitae, life. So the tree of life, you can see how it branches out. It looks a bit like fan coral to me. Okay, let's do one more incision here. I'm going to cut the frontal lobe off and make a cross section, and so we should be able to make a nice cross section drawing here and see the difference between the grey and the white matter. Notice that the grey matter is more on the exterior side of the brain, the white matter is on the interior side, on the inside. And it's to do with how it is myelinated, the myelinated sheaths or not, for the protection of it. So it's really quite pretty, don't you think? You can also see quite clearly these gyri and the sulci. You can see how deep these sulci get, to see how we can have increase in surface area.